This is a seed burial study run by a PhD student, Hannah Lindell. She's looking at the weed seed degradation of 4 different species. And so the idea behind this study is to look at the different impacts that a cover crop, annual or perennial in the case of the living mulch, what are the impacts on weed seed degradation? Since we have a perennial living mulch, we have something green on the soil surface year round, whereas here we have different nutrient dynamics that are happening and nutrient releases in something like a crimson clover versus our living mulch. And in the case of our cereal rye, we have very large root systems and things like that that could affect soil microbes and even insects that might be on the soil surface. We found that we see a lot more fire ants predating weed seeds in some of these plots. And so these weed seeds are buried at 4 different depths. We have some that are on the soil surface, which is what you can see here, protected by this wire mesh. And so the idea there is that we would see variability not only between the cover crops treatments, but also between the burial depths.
Hi, my name is Hannah Lindell. I'm a PhD student in the weed science program here at the University of Georgia, Athens. Today I'm going to discuss to y'all a seed burial study project. What we wanted to do is we wanted to look at 4 prevalent weed species in Georgia production and kind of understand exactly what's going on below the soil surface with those seeds. And those weed seed species consisted of crabgrass, broadleaf singlegrass, Palmer amaranth, and morning glory. And then we also wanted to look at not only what those cover crop systems effects had on those species, but also the depths. And the reason why we decided to go with those 4 depths is because we wanted to best represent the type of tillage and land preparation methods that growers use in the state of Georgia. Of course, our 1 and 0 inch would represent minimal to no-till. Our 4 inches would represent using a cultivator, which goes between 4 to 6 inches under the soil. And then, of course, our 18 inches represents using a moldboard plow to completely till up and fold over that soil. The duration of this trial goes for 3 years. What we hope to see within this study is, especially with our living mulch system, we have more seeds degrade over time. And this can be because with our living mulch system, we have a continuously growing plant or vegetation. And so our soil is going to have either higher microbial content in it, greater possibility, which leads to greater possibility of that seed degradation. Whereas with our bare ground system, we're hoping to see less seeds degrade over time, but more seeds emerge because there's a better condition for those weed seeds to germinate over time because there's less competition with other weed species and crops as well. We also hope to see that with our seeds that are buried at either the soil surface or 1 inch below soil surface are going to have more degradation over time and not as much longevity. And this is because there's a better chance they'll be exposed to their conditions around them. So they'll either degrade over time or they'll germinate within their bags. And so we'll have fewer seeds to count within those seed depths, whereas seed buried at 4 inches or 18 inches under the soil surface, we believe that there'll be more seeds will either go into dormancy, fewer seeds will degrade over time, and if they were in a scenario where our moldboard plow is used and they're brought to the soil surface, we'll have more seeds germinate with those depths. And one of the things that we do to look at the actual longevity and viability of these seeds is after we exhume them at their time periods, we put them through a process which is called a tetrazoium test. And essentially what a tetrazoium is it is a chemical that interacts with carbohydrates in the seeds, and it essentially will stain the seed spread if they have a lot of carbohydrates, meaning that those seeds will likely produce a plant and therefore they hold on to their longevity or they do not degrade within those systems. Seeds that don't, they don't die, they don't change color, and so we then label those seeds as non-viable. So once we exhume our seeds from the soil, what we do is we then place them into our freezer here. And this essentially what it does is it allows us to preserve these soil samples long enough for us to be able to pull them out the freezer, thaw them, and then put them through a calcium chloride solution, which allows us to separate the seeds from the sand, which is within each one of these bags. So for each depth, we have 4 bags, which represent our 4 weed species. So we have our pomegranate bag, our crabgrass bag, our morning glory bag, and our broadleaf sandalgrass bag. So here's what one of those bags look like once it's pulled from the soil surface. So these bags consist of 20 grams of sand as well as 100 seeds of whatever weed species is in these bags. Like I said, palmer, crabgrass, morning glory, broadleaf sandalgrass. The reason why we put 20 grams of sand is essentially just to reduce the amount of contact between seeds. Once we separate each one of these weed species, we then put it through a 10% Clorox cleaning solution. And the main reason why we do this is we want to reduce the amount of bacteria or microbes on those seeds so that when we end up putting it through its tetrazoium test, there is no extra living organisms within that sample that can then alter the actual viability and testing of those seeds. Here's just kind of what those look like in the vials. So next, once the seeds have soaked in the DI water in their respective vials for 24 hours, we then transfer the seed onto petri dishes with filter papers, and then we conduct a tetrazoium test on them. Once we've added the tetrazoium solution, the petri dishes are then placed into the refrigerator. They'll sit there for 24 to 48 hours, and the solution will then be imbibed by the seeds. Once they are fully imbibed, samples are then pulled out and a viability versus non-viability test is conducted. Essentially what that looks like is we separate seeds that have completely been stained the color red or a pink to purple color by the seeds that have not. And seeds that have been stained are labeled as viable, meaning that they will eventually produce a plant at some point, or non-viable, meaning that they will not produce a plant if they are presented to the soil surface or pulled up at 3 months after. But so far, especially with our 3-month, we have seen that with our 1 inch to soil surface, we actually have a high percentage of weed seeds either degrade or kind of are not viable after a tetrazoium test, whereas a lot more of our seeds especially are 18 inches on their soil surface, they have higher viability, so they are stained red with their tetrazoium, as well as although with our morning glory seeds, we've actually seen that under our living mulch systems, whether it be at 0, 1, 4, or 18 inches, we actually do see pretty good degradation of those weed seeds, especially at 3 months.